Okay, so hello everyone. Thank you so much for joining us and welcome. My name's Rachel Taylor. I'm one of the Principal Scientific Program Managers at EMQN. EMQN is a quality assurance provider in genomics, so we provide external quality assessment services to over 3,000 laboratories in 82 different countries all over the world. And we're also a community interest company, so we provide educational services to the community as well. And we wanted our ESHG workshop this year to celebrate 25 years of HGVS. So I'm sure most of you in this room know that HGVS is the internationally recognized standard for the description of sequence variants. And we've got two really exciting speakers presenting for us today. They're both providing presentations on really important topics in this area, so we hope you'll enjoy the session. So after my introduction, both Ivo and Alex will give their talks, and then we'll have a short Q&A session at the end. But we've got the room booked for an extra 30 minutes, so there'll be an opportunity for you to network and speak to the presenters directly if you want to continue any of the discussions after the session today. So just to let everybody know, this workshop is being recorded, as will the Q&A session. We just wanted to let you know that this recording will be disseminated to our community after ESHG has finished this year. Okay, so I'm really pleased to introduce to you our first speaker, Ivo Fokkema from Leiden University Medical Centre. So Ivo's been working for over 20 years on the Leiden Open Variation Database project, which powers the largest network of curated gene variants in the world. And in more recent years, as LOVD's PI, he's been focusing on improving data sharing between systems through improving standards, data formats, automation, and data validation. As an active member of several committees of the Human Genome Organization, Ivo is involved in the HGVS Variant Nomenclature Committee, Reporting of Sequence Variants Working Group, and the HUGO Forum. And he also chairs the HUGO Gene Disease Specific Database Advisory Council. So with that, I'd like to invite Ivo up to the lectern. Thank you.
All right, thank you, Rachel, for the introduction. And welcome, everyone. I hope you're enjoying the snacks. So yes, I will be talking about validation tools for HGVS variant descriptions. But first, I will need to convince you why you actually need them. So what you may not know about the HGVS nomenclature, indeed, it's been 25 years now that it's first published, but the first work that started the nomenclature actually started already in 1993. And back then, the guidelines were based on all the descriptions that people were using already then in the literature. So that means that the initial guidelines were actually already quite diverse. This, of course, was meant that people could easily switch to the HGVS nomenclature because it was so similar to what they already were using, but it did make the nomenclature very diverse. It's primarily aimed for humans rather than software, so it had to be readable for humans, but that doesn't always make it very logical for software to process. And it has been updated several times, so sometimes something that used to be valid description is actually no longer a valid description. And it was extended over time to allow for multiple new variant types to be included. And this makes the HGVS nomenclature very powerful. There are descriptions that you can only describe in HGVS nomenclature and cannot be described in other variant standards, but it does create some challenges. So this was highlighted also when we analyzed 1 million database submissions to the LOVD software, as well as going through 5,000 published papers that focused on retinal diseases. And one of the most common mistakes that we found is that descriptions used older nomenclature. So for instance, C.100del-A means that there's a deletion on this position 100, but actually it's not necessary to mention that base. It's actually that the guidelines say that you should not mention that base. But people are still often using the older nomenclature where this base was mentioned. There's also typos if people manually create the descriptions. So for instance, the minus is normally used for a offset in an intron. So literally C.100 minus 200 del means a deletion of 200 bases into the intron before position 100. But actually in this case, the author of that paper meant that an underscore, so it was like a range of, well, with this example, 100 bases. Journals sometimes format variant descriptions. So for the PDF, they sometimes include spaces in the middle of variant descriptions, or they use special characters that in the PDF look exactly like the way it should look, but if you then copy that variant description to paste it, for instance, in a database, something else entirely pops up. Very common errors also in papers is not to mention a reference sequence. So then actually I saw it a lot in the posters here as well. You see a gene symbol on the variant description, but it doesn't actually specify what transcript was used. So sometimes that means that you actually have an ambiguous description and you wouldn't actually know if that variant is also found in one of your patients, for instance. It can get worse. The DNA level variant can be removed. So then you just have a gene and a protein change. Well, there's multiple DNA changes that can actually cause this protein change. So you don't actually know what variant they were talking about. It can get worse. Maybe the protein change is actually just a prediction, and maybe that's not at all what actually happens. So you really always need to make sure that the DNA description is included. Sometimes HGVS nomenclature was not used at all, and there was just a piece of text that we needed to understand to see what kind of variant was actually in there. So most database searches rely on text matching. So even when you have a description such as C.100 del A, if you search in a database that uses the official nomenclature, you will not actually find C.100 del because you included more information, and that does not include in a database. So before you search databases and before you pull in annotation from other sources, make sure that all your variant descriptions are valid. Now, you may think that this does not really apply to you because you may use this, descriptions that come directly out of your sequencing pipeline. Well, unfortunately, there are a lot of scripts out there and programs that generate HGVS expressions, but that doesn't mean that these are also valid. When I searched on GitHub, which is a platform for open source software, I found 142 software packages that have the term HGVS in their name or their description. They may not all generate HGVS variant expressions, but they certainly a lot of them do. And then, of course, there's all the commercial tools that are actually not included in GitHub, but that do produce HGVS expressions. Even well-known software has problems. So, for instance, Ensemble Fab doesn't always produce valid descriptions, NOVAR, and even technically speaking, if you look at ClinVar, all the protein descriptions are actually, technically speaking, invalid because these are actually predictions, but they're marked as if they're not. Sequencing pipelines produce VCF files, but VCF files, they left align all the variants, so they want to make sure that all the variant descriptions are basically using like the first bases that changed. But the HGVS nomenclature actually specifies that you should shift the variant description as far 3' as possible. But that means that lots of sequencing pipelines actually create HGVS expressions that are in the wrong place. And this was shown to us in a very interesting way when we collected data from all the Dutch diagnostic labs, and there was one specific deletion that got submitted to us in 16 different descriptions. So 16 different HGVS descriptions that were, well, 15 of them were wrong, one of them was correct. And there was one lab even that submitted the same variant 12 times to us. So they classified the same variant 12 times simply because their sequencing pipeline generated 12 unique descriptions for the same deletion. So even if you use a sequencing pipeline, generating HGVS descriptions always requires validation. But what about liftover? Maybe you are using liftover to move your HC19 data to HC38. And maybe you expect this to be simple, as this example, where you simply change the position, but the base does not change. But it's a new reference sequence. That means sequence has changed. So what if a variant actually disappeared because it's included in the reference? Or, can get worse, you have a deletion insertion that actually changes in a duplication. Or it can get even worse, you have a deletion insertion that on HC38 is a deletion, but on the transcript it's actually an insertion. But liftover tools, they do not check the sequence. So when you use liftover tools for your HD19 data to HD38, you will have problems. You can use this for CNVs, where it's just really about the positions, but when you want the actual sequence to be validated, you cannot use liftover. Also, remember the variant that you actually observed. So if the data is generated on HD19, make sure that you label this data as the original observed data. Anything else you create after that is annotation. You need to make sure that the original data is labeled as such, because if you map this to transcript or HD38, and then later you will maybe map it to the telomere-to-telomere reference sequence or something else, you may induce even more errors. Make sure that you label the original description, your actually observed variant, as the original one. Now, I hope I've convinced you that you need to validate the variant descriptions that you have, but now how? So the HGVS nomenclature website lists 5 different software tools that you can use, and I'm going to talk about 3 of them, because these 3 have an online tool, so an online version for you to use, a batch tool, so where you can submit a larger number of variant descriptions, and an API for sequencing pipelines, so for bioinformaticians to directly communicate with the software. So these 3 tools are Mutilizer, Variant Validator, and the LFD HGVS syntax checker. Each of them have unique capabilities that no other tool has, and I will talk you through them. And to make it a little bit easier, so you may actually need like multiple of these tools because they may have features that you need. But to make it a little bit easier for you, the last 2 software projects have started working together, so then it's actually easier for you to use them both all in one go. So first I'm going to talk about Mutilizer. This is the first HGVS nomenclature validation tool and developed in the LUMC in Leiden, the Netherlands, and focused on a broad range of unique sequence level tools that are not found anywhere else. I'm going to give you a few examples. So the algebra feature can tell you how 2 variants relate to each other. So you may have 2 descriptions that are actually the same variant, but you can also, for instance, want to know of a certain substitution, actually part of a deletion insertion. And the algebra feature can actually tell you whether or not these 2 variants are related to one another. The back translator is very useful if you have a protein change, but you do not know what DNA change has caused this. So the back translator can predict which DNA variants are most likely ones that have caused this protein change. And then there's the description extractor that generates variant descriptions based on 2 sequences. So it's very useful, for instance, if there's a new version of the transcript that you're interested in, you can insert the sequences of both versions of the transcript, and you can actually see what the differences are between them in HGVS expressions. The description extractor is also used internally by Mutilizer to make sure that any given variant, no matter how you input it, it always gets the same reply. So it always has the same outcome of the tool. That does bring some disadvantages with it. So for instance, the original context is lost. So if you submit an insertion, and according to the nomenclature, this should be a duplication, Mutilizer will of course change this. But because the description extractor doesn't know anymore that you used an insertion to create the change in the sequence, it doesn't give you any feedback. It doesn't tell you you shouldn't have used an insertion, this is a duplication. So it's actually kind of difficult then for you to see whether the change that Mutilizer created is a problem of your input. Did you do something wrong? Or is it just that the tool has optimized and changed the variant description to something else? Because it does actually use one output only, but it doesn't always mean that that is the only valid result. So for instance, a duplication, when it's in a repeat region, you can also use the repeat syntax to indicate that the sequence has expanded. These are both valid descriptions, but the description extractor chooses one of them as the output. That doesn't mean that the other one is invalid. So it is a bit difficult for you to see which one is invalid because you didn't follow the nomenclature, or whether it's simply that the description extractor has a different preference at that moment. The mapping to transcript is slightly non-conventional and actually needs multiple steps. So when you when you put in a genomic variant description into Mutilizer to get a mapping on the transcript, the reference sequence that's used is actually an NCNM. I'm not sure if you can see the pointer, but this here is an NCNM. And this numbering does not match the NM numbering. So some users think that this is then actually already the mapping to the transcript, but it's not yet. It's like a sort of in-between version. In this case, this deletion insertion is actually already incorporated in this reference sequence. So on the NM level, that variant actually doesn't exist. But Mutilizer initially does give this description that looks like a transcript level description, but it's actually not how the variant should be described on the transcript. You need an extra step for this. This NC-NM structure is a bit complicated. The HGVS nomenclature doesn't actually clearly define what it means, how it should be used, how the sequence should exactly look. They do tell you that you are required to use it when describing variants in the intron, but they don't actually say how exactly it should be built. And that resulted in Mutilizer and Variant Validator doing two different things for what this NC-NM actually means. Okay, Variant Validator. So that one is developed by the universities of Leicester and Manchester in the United Kingdom. And it was designed as a more accurate alternative to Mutilizer, but that was mostly compared to version 2. It focused on user friendliness and handling alignment gaps in reference sequences. And some of the advantages are that it does give you informative messages about your input. So if there's something wrong with your input, it will always tell you. And it handles alignment gaps from genome to transcript and genome to genome in one request. So Variant Validator's implementation of this NC-NM construct, the numbering for the NC-NM always matches the numbering on the NM, so those are actually like usable. And it provides a downloadable PDF report and some additional services to get some information about the gene or transcript, mapping of the transcript on the genome, etc. But also Variant Validator has disadvantages. For instance, the repeat syntax. This is an example of an expanded repeat from 3 CIG units to 4. It only handles this as input, so it does understand what it means, but in this case, the output will be a duplication. It doesn't actually use the same syntax as what you put in. Also like a complex insertion, so where more than one things happen in one go. In this case, there's a duplication and there's an insertion of a different repeat unit in this area. This is not understood by Variant Validator, unfortunately. They also sometimes provide a lot of information on one page to make sure that you get all of the data, but it does make the screen look a bit cluttered, so maybe you can't really find everything that you need, but sometimes you need to scroll to get more information. They did recently integrate the LFD HGVS syntax checker for additional feedback. So whenever they don't understand your input, they will submit it to the, it's kind of a long name, sorry, LFD HGVS syntax checker. And then this tool will actually provide you more feedback about what might be wrong with your input. So I'm not going to repeat the name of I think I need a better name for this software. So that's developed by me. It originated from the LFD software, that's why it's in the name. Also developed in the LMC in Leiden, and it focuses on supporting the full HGVS nomenclature. So there's lots of very rare variant descriptions out there that are not really implemented in the sequence level validators, and this is just a syntax checker, so it looks actually just at the syntax. And we also looked a lot at all the common user errors, so this is the research that I mentioned, that we looked at 1 million variant descriptions in the database, and we looked at the 5,000 papers. We really looked at what people were often doing wrong to see how we could interpret all of these and still create the correct descriptions for this. So some of the advantages is that it allows processing variant descriptions without reference sequences. So if you have a paper, for instance, and there's lots of DNA descriptions in there, but the reference sequence is somewhere in the material and methods or so, that's not a problem because this tool just checks the syntax, so it can check all the DNA descriptions, even if there's no reference sequence. And also supports descriptions that sequence level validators simply can't validate, like variants with the uncertain position or variants with the uncertain sequence. All of these can still be validated on the syntax level. So it supports 98.5% of all the DNA variant descriptions that are listed on the HGVS nomenclature website. The only thing that's actually missing is stuff that isn't very clearly defined on the HGVS nomenclature website. So that's something that we try to fix on the website so that actually then the tool can validate all the descriptions as well. So really the aim is to be able to support the full nomenclature, no matter how rare the variant description is. Like I mentioned, also lots of common user errors and alternative syntax like VCF type syntax or SPDI is all interpreted and translated into HGVS. And because it only does a syntax check, it is very fast. There are, of course, disadvantages. So for one, because it's a syntax level validation, you still need sequence level validation, and this is validation only. So we don't actually do any mapping or protein prediction. For this, you really need the sequence level validator, so mutilizer or variant validator. But to fix at least this problem for our users, we integrated variant validator into the online tool. So when you use the online tool, you can just link the option, I will show you in a bit, and then it does direct sequence level validation as well. But of course, this only works for variant types that are actually supported by variant validator, but the tool will indicate this for you. And it, of course, only works if you have the reference sequence attached as well. We are willing to also get the RNA and protein descriptions validated. This is still work in progress, but I hope at the end of this year, also RNA descriptions and protein descriptions can all be validated using this tool. To just have a quick look at what it looks like. So in this case, I inputted a VCF style variant description. I then click on the button to have the validation start, and it actually recognizes what this meant. It gives you a confidence as well. So in some cases, it's not fully confident of the correction. In that case, it will give you a certain confidence value. In this case, it's highly confident that this is actually what you mean. So you can just click on the description that's given. So it will automatically resubmit and update your description. And in this case, it says that the syntax is valid, but that you still did not check the sequence level. So then we have a little checkbox for that. So you can click on the checkbox and resubmit the variant description. And then in that case, it seems that this insertion should actually be described as a duplication. And also here, you can just simply click on that link, and it will update your description and now give you a full green result, because now it's also validated on the syntax level. Sorry, on the sequence level. So in this case, you don't actually need to use both tools. You can just use both tools in one go. We do have an option for the batch check. So on top, there's a little tab that allows you to submit a whole list of variant descriptions. And on the right, there's a download option that gives you a text file with your input and all the output that's generated by both us and variant validator. So you can have a look at what you need to change if you need anything. So just some key takeaways. The database searches rely on text matching most of the time, so make sure that you use fully correct descriptions. When generating HGVS variant descriptions, even if you generate them from your pipeline, always make sure that you have them validated. Don't use liftover for variants. CNVs is fine, but not for variant descriptions, because looking at the sequence, sometimes the variant description needs to be changed completely. And when mapping a variant, always remember which variant you actually observed. That is your original data. Make sure you go back to it if you need to pull in more annotation. When using Mutilizer, make sure that you actually map to the NM. So the NZ-NM, the numbering is not compatible to the NM's numbering, but variant validator does do it all in one step. So there you will actually get the mapping from the genome to the transcript and genome to genome in one step. And the LFD HGVS syntax checker validates all rare variant types and alternative syntax in one go. That was it. Thank you for your interest.
Thank you, Ivo. That was a really interesting talk. I'm sure there's lots of questions, so I recommend writing those down so you remember them for the Q&A session at the end. Okay, so it's my great pleasure now to introduce to you Dr. Alex Wagner, who is an assistant professor at the Institute for Genomic Medicine at the Nationwide Children's Hospital in Ohio. His research focuses the development of computational standards and tools to improve the scalability of genomic variant interpretation, with a focus on recurrent cancer variants such as gene fusions. He's the co-lead of several community variant interpretation initiatives, such as the ClinGen Somatic Cancer Program, the Variant Interpretation for Cancer Consortium, the Genome Knowledge Standards Workstream of the Global Alliance for Genomics and Health, and the HGVS Variant Nomenclature Committee. So with that, I'd like to invite Alex up to the lectern.
Thank you for that introduction. So it's my privilege to speak to all of you today about work we've been doing across several of these community initiatives on the representation of gene fusions. This is a challenging problem because the way we talk about gene fusions is highly variable depending on context in the biomedical literature and clinical reports and the outputs of gene fusion prediction pipelines. We end up with all sorts of different notations that span everywhere from genomic DNA breakpoints down through the notion of entire families of genes and the relations that they have when in fusions to cause different forms of disease. And so a nomenclature that can specify these varying levels of granularity in a consistent way is a major challenge. So to address this, we developed a cross-consortium working group to come up with a standard, consistent information model, definitions, and nomenclature to define gene fusions. The groups involved are shown on this slide, and this is really how the project got started. But we're also very well aware that when you're talking about developing new standards, there's this risk that you are effectively making things worse by introducing yet another standard that people need to recognize and use and be able to align with the existing ones. So up front, our goal here was not to develop something that was breaking from existing community practices, but rather building upon them. So in light of that, as we developed these recommendations, one of our key steps was to align these with the nomenclature committees of the Human Genome Organization, or HUGO. And this includes representation from the HGNC, or the Gene Nomenclature Committee, the HGVS Nomenclature Committee, and of course ISCN. And so working across these groups, we came up with a consensus set of recommendations, which we'll be talking about today. But first, a little history. So in 2021, there was an HGNC gene fusion nomenclature publication produced by, of course, the HGNC, where the recommendation was quite succinct. It was effectively using gene symbols with a double colon delimiter in order to specify a description of a gene fusion at the gene level. So in this case, BCR : ABL1 as an example of a gene fusion event. Now, the nice thing about the syntax is that it aligned with prior community recommendations, the ISCN guidelines for genomic rearrangements, the HGVS nomenclature for what they called fusion transcripts, and then guidelines for gene fusion descriptions that existed in other organisms like Stolfi et al. Describing gene fusions in tunicate genomes. This short-form descriptive syntax, however, of course, is quite limiting in what it can describe. There's no way of using staple gene identifiers within that syntax. There's no distinction between what we consider regulatory or chimeric transcript fusions, nor are you able to differentiate between chromosomal events and gene fusion events. And it also doesn't support, any form of sequence level description, but of course they do refer to HGVS and ISCN for those applications. So one of the things that, one of the challenges about this is that its applications are therefore limited. So it's designed specifically for shorthand, for use in publications, not for clinical reporting. And it's really focused around what HGNC does, which is how do we name genes? And that's really the way that they thought about this. And one of the primary challenges of that approach is that they didn't really start from a working model of what a gene fusion is. Rather, they were thinking about it of how do we name these types of events. And without a working model of what it is, it's very hard to then say what content belongs in your information model and how do you build a nomenclature around it. In HGVS, this is at the beginning of last year. This is January 1st, 2024. This is what fusions looked like in the HGVS nomenclature. We had a fusion transcript in our glossary, and we had this example of fusion transcript, which existed buried down in the examples and notes of the splicing recommendations on the HGVS nomenclature. And this is effectively what people would use then as a model for how they'd represent fusions in clinical reports. So we wanted to try and build off of these foundational roots from the HGVS and from HGNC to build a more comprehensive system that was well-defined, and we developed the gene fusion specification. Now, this work is available at fusions.cancervariants.org, and it contains an information model, a nomenclature, and a set of definitions that we're going to go through in the rest of this talk. So to begin, we started with the definition of what a gene fusion is. And there are really two broad classes of gene fusions that people typically think about. There's your canonical form, where you have two chimeric transcript that forms from two different protein coding products. But you can also have regulatory fusions, where you have a promoter element or a nearby enhancer element that you rearrange another gene towards, that then affects the regulatory behavior of that gene partner and produces some phenotypic effect. And so our definition of gene fusion encapsulates both of these forms, but we have different ways of specifying these different classes of gene fusions, as you'll see in a minute. Importantly, we wanted to make it very clear about how gene fusions are distinct from rearrangements that drive them. They can be inferred from DNA assays, and this is captured in the specification information model. But importantly, they are distinct from these rearrangements, and you can have gene fusions that are not involved with rearrangements at all. Also, I just want to note that while many gene fusion callers will also provide information about internal tandem duplications, these are not gene fusions. They just don't fit inside the definition of it. That isn't to say that some of the same structures aren't applicable, only that this is outside the scope of what that definition is. And it's because they do not involve, of course, two or more genes. So if you were to look at HGVS today, we've updated what we consider gene fusions to be. So we've adopted a number of different definitions from the VIC project about chimeric transcripts, read-through transcripts, gene fusions, regulatory fusions. All of this vocabulary now exists inside HGVS. We've also deprecated the fusion transcript definition in favor of a new structure called a join transcript. So this is a way of being able to represent the adjacency between two transcript segments without any sort of added baggage of is this a gene fusion or isn't it. It allows you to simply define the structure of the molecule that you're observing. And this is what it looks like. So we have a formal syntax and examples. This has now been split out from that splicing page. It has its own page on the HGVS recommendations site. This exists under joined transcripts under the RNA section. And what it allows us to do is provide a formal structure and grammar that allows us to specify kind of the two different segments and their adjacency, as well as an optional linker sequence that intervenes. So you can go to the existing HGVS nomenclature site for more details about this, more examples, and application notes. So there are some strengths and limitations to the current model. It is a consistent syntax with the prior examples that existed on that splicing page. So you don't need to change anything if you've already deployed your fusion pipeline to use HGVS-like examples from the splicing recommendations a year ago. This should be consistent with that. It also supports two transcripts, which may be for a gene fusion, but might also be just multiple transcript segments from the same gene, and an optional linker sequence. So it's a useful way of bridging these concepts. However, there are some limitations, and one of these is that many clinical labs want to be able to report based off of exons in transcripts, and this nomenclature does not support that, nor does it support regulatory fusions or 3 or more partner fusion events. So in order to address these concerns, the HGVS took a look at the work done in the VIC specification and endorsed it for use for those applications. So HGVS, much like it did with the HGNC recommendations, simply recommends the use of the VIC specification and its nomenclature for representing fusions that have multiple partners that you want to represent using exon-based representation, et cetera. And we'll talk a bit about what that nomenclature looks like now. So we started with an information model that has the structure of gene fusions. So here you can see a number of concepts we took directly from HGVS. The notion of transcript segments, where you have segments defined by 3' and 5' boundaries that together constitute the different components of a chimeric transcript. These include the notion of its exon number and any offset, either into the intronic or exonic space, and also an associated genomic location to help you orient this and reduce errors when it comes to multiple possible alignments that might exist between transcripts in the genome. Now, the way it works is we have a number of different types of components. I just talked about transcript segments, but we also have both gene level as well as regulatory level components that can be mixed together in order to create fusion events. And we have a workflow for how you can assemble arbitrarily long fusion events using these components. So an example of walking through this, you might have a simple junction between a 5' fusion partner and then a double colon, and then a 3' fusion partner, and then the end. That's kind of your simplest and I think most common form of a chimeric transcript fusion. But you can also have regulatory fusions that involve regulatory elements, and for that we actually had to define a little mini nomenclature because this didn't really exist. And this includes the type of regulatory element you're observing. Where that exists is optional. This can be either by reference, by location element, or you can even associate it with a gene, and then that constitutes that part of your regulatory nomenclature. And then that can be appended onto a 3' partner that is regulated. Now, each one of these different components, all those different boxes, have their own syntax for transforming the information content of that component into an actual nomenclature string. I'm not going to go through all the details here, but this is available on the site, so you can learn about the specification, about how these get applied together. But I'll run through a few examples, which I think might be an easier way to kind of describe how this works. So earlier I mentioned BCR-ABL1. This is the example used in the HGNC recommendations. Our syntax follows the same recommendations as HGNC for gene-level events, but does optionally allow you to specify gene identifiers. Now, the emphasis here is on optional. There are some communities and there are some formats that don't work nicely with having colon delimited Curry identifiers as stable IDs associated with genes, and for those applications, of course, we don't recommend you use this. But there are other applications where this is very valuable, so it's an optional part of the syntax. Another thing that we provide is a distinction first between unknown partner fusions. So this is like a fish break-apart assay where you have a probe that kind of monitors if the ALK gene has been rearranged into two different components. And when you see that type of signal, as shown on the screen here, that's indicative that you likely have an ALK fusion where the 3' ALK kinase domain has been fused to a partner that's likely driving the cancer of interest. In contrast, we also have this V syntax describing a variable number or a large number of known partner genes associated with another gene. So in this case, V : KMT2A is an example of one that we extracted from the WHO classification of hematopoietic tumors. That allows us to specify this is a known recurring fusion partner that has a number of different associated partners. So this is a useful thing to put into databases or knowledge stores that allow you to basically associate any KMT2A fusion with such a fusion you've observed in your own assay. Okay, it also provides, as I mentioned before, a structure very similar, analogous to the HGVS structure for transcript segments. Here, though, it's represented using exons, so we have this E-dot syntax that allows us to say from the beginning of exon 1 through the end of exon 7 in the left transcript with a linker sequence, and then another transcript on the right that goes from exons 3 to 9, as shown in the blue. And so these different structures allow us to then kind of represent the main components of the structure of this fusion transcript in a more compact syntax. Now, I will note that this syntax, much like HGVS, requires the use of reference sequence identifiers. And this is because in order for us to resolve exon numbering into coordinates, you must know which transcript structure you're talking about, so the transcript identifiers are required and essential. The other thing this supports is a more succinct form, a shorthand syntax that allows us to specify only junctions. So often when you're doing assaying of gene fusions, you'll find the junction where you've sequenced across, but you won't actually know if it goes all the way out to the end of the transcripts. And you really just want to specify the junction. For that, we have this shorthand form where you say the exon number at which the junction starts, and then the exon number for the 3' end of the junction. And so you're just talking about that bridge and nothing beyond it. And then I also mentioned offsets briefly. Let me visualize what that looks like. So here we have a +2 offset from exon 1 and a +5 offset from exon 8 in the partner gene, and you'll note that that positive offset on the 5' end is going into the intronic sequence, and a positive offset on the 3' end goes into the exonic sequence. So those offsets are always with respect to the reading orientation of the transcript. Okay, and then finally, regulatory fusion. So here's a well-known regulatory fusion, which is IGH-MYC. And in this case, we've simply appended from our regulatory fusion nomenclature this notion of this being an enhancer element, which is the _E on reg_E, at the IGH gene locus associated with MYC. And this gives us our regulatory enhancer-driven event. Okay, so that's a high-level overview of the nomenclature, the information model, and the work done to build a comprehensive and consistent cross-community specification for gene fusions. It's exon-based, it's compact, it provides all the necessary information to be meaningful in the clinical setting. This has been aligned with all of the HUGO nomenclature committees, and it's a formal recommendation from HGVS. So with that, I'd just like to acknowledge all the different groups that worked on this, the co-lead of mine on this project, Gordana Rasa, and of course the funding institutes that help support this effort. I think with that, I'll pass it back to Rachel.
Thank you, Alex, for that very clear explanation of what is actually quite complex nomenclature, so thank you. Does anyone have any questions? You've each got a microphone at your station. If you've got a question, you can just press the button and speak.
I have a question for you. The Global Alliance has another variant representation model. What are your thoughts about that? No, Ivo.
Actually, it could be that Alex has a much better understanding of. You mean verse, right?
Yeah.
Yeah.
Right.
Actually, Alex has a much better understanding of verse than I do. But I think it's very important that it ends up with one description for each variant. So that is the best part of it, that there will only be one description for each variant, because that is still a problem within the HGVS nomenclature. Like, I mean, the repeat syntax is just one description where you can describe an expansion of a repeat in multiple ways. But I mean, for instance, if you look at the protein level descriptions, it gets even worse, because you have the 3-letter amino acid code, you got 1-letter amino acid code. I think in total you have like 5 different ways to describe a frame shift. So that is what verse does really well. The problem is, however, that it will always require something for humans to use and for humans to look at. So I think it will never, HGVS nomenclature will never be replaced by verse, but verse is a really good tool to compare variant descriptions in a way that you will really know the difference, like if it's the same variant or not.
Yeah,
I hope that answers your...
I mean, it's really hard if you develop a variant database that you search for a primary key, which is kind of independent of reference genome, for example. So it's not an easy task, and I think it's a very important task, and I'm still wondered why this is not really solved.
Yeah, so I actually think that for database searches, verse is a great solution. But then what if you still want to search for variants in a certain codon? Or what if you want to search for variants in a certain range? All of this will still need additional nomenclature. But for looking for a full variant description, to really look for an exact match, VERST is the best option for that, yeah. And I really hope that more databases will start incorporate it. Me, myself, I also haven't incorporated it yet, so.
Okay, so it's not offered in the conversion tools you mentioned, it's not
Currently not. So,
okay.
So for me, the focus at this point for me, for the syntax stack, is to be very fast. And to generate the first description, it actually needs to look at the syntax, sorry, the sequence as well. But once variant validator starts implementing VERST, then we can simply pull in that information from them. So we kind of divide our work in such a way that they do all the sequence level validation stuff and we do the syntax validation stuff. So once variant validator actually incorporated VERST, and they want to, it's on their list, then also LFD can start using it within the database and within the tooling.
Okay. Thank you very much.
You're welcome.
I'm going to just jump in there. So I didn't talk about it at all in my talk, but VERST is actually something that I have worked on pretty extensively as part of GA4GH. And I will say that, you know, I agree with everything Ivo said in characterizing it and its uses. VERST 2.0 was released in January of this year and includes the ability to include in these VERST objects the descriptions that come from different nomenclatures, not just HGVS, but also SPEEDY, Nomad style, VCF, et cetera. And there are conversion tools that do exist, and we've used these at scale. So the tech stack, so Variant Validator, is built on a HGVS package that is maintained by a community called Biocommons. And that same HGVS package is used for the VERST Python, the reference implementation for Python in the Global Alliance for Genomics and Health. So the translators that work there they are deep in that tool stack. They exist and we use them in GA4GH, although they exist over in Biocommons as a separate community. And I think the thing that's lacking is those updates to HGVS that include this conversion over into the Variant Validator stack. So there was a forking that occurred years ago between HGVS and HGVS VV, which is the Variant Validator version of that software. And there is an active effort with Variant Validator to bring those packages back together. So I think when that occurs, they're going to get a lot of this tooling for free.
Okay, thank you. We've got another question at the back just there.
Hi, my question is for Ivo. So how do...
Oh, Mariana, a bit closer.
Oh, sorry. Now can you hear me?
Yes.
Yes. So for Delvin's nomenclators, I see that the HGVS guidelines now are more clear, but I see a completely different output in both the tools, for instance, Mutualizer and Variant Validator. How do you see that this is solved in the future, or we would still need to do it basically manually because this will not be possible to apply? Thank you.
So do you mean specifically, when you see there's different output, do you mean specifically that you get like a different mapping? So for instance, from genome to transcript? Or is it that a certain variant description is given differently?
The variant description. Imagine if we have a SNP, and then 2 nucleotides, and then a deletion. If you go to Mutualizer, it will show you as a SNV, and then a deletion. But if you go to Variant Validator, it will show you as a delins.
Yeah, so that's actually, that's a great example of where this description extractor is causing the difference between the tool. Variant Validator will stay close to what you put in. So if you put in the deletion insertion, Variant Validator will keep it a deletion insertion. But Mutualizer, since they use the description extractor, they will see that in reality, this is more efficiently described as 2 separate variants. Because according to the HGVS nomenclature, when variants are further away than 1 nucleotide, you should actually split them. So in reality, in this case, Mutualizer is actually correct. These are 2 variants. They are 2 variants on the same allele, but there are 2 variants. variant validator will simply look at, you gave me a deletion insertion, I will keep it a deletion insertion. So this is an example, and it's also related to the question about verse, where there are still two descriptions around that actually mean the same variant. And in this case, HGVS nomenclature is sort of clear about that there should be two variants, but it still says on the website, if it helps for the implementer, they can still be grouped together. So the HGVS nomenclature still says that you're allowed to merge them. Even though they recommend you not to, you are allowed to merge them. But then this is a problem with searching in databases. So that's also why I think in that case, verse would be a much better alternative to really make sure that all the variant descriptions will have the same description, because people will still want to read something specific to their needs. So some people will want to describe this as a deletion insertion. Some people will want to describe it as two separate variants, but you still need to find them both, and verse can help with that. So I don't think you will need to do this manually, but it will require you a bit more of your patience, I'm afraid. But thank you for your question.
Thank you. Any more questions? Okay, with that, I'd like to say thank you to the presenters for the really interesting discussions today. If you'd like to see more of EMQN's work at ESHG, we've got the QR code just up there. And please come and see us at booth 358 if you'd like to talk to us more about EQA. And also, please help yourself to the food that's available at the back of the room before you leave today. And any further questions you've got for Ivo and Alex, I'm sure they'd be happy to talk with you. And we've got the room until 6:30 if you'd like to do that. So thank you very much, everybody.